Hello, I'm Kelly Cassavale, the director of the Agriculture, Food, and Nutrition Evidence Center in Fort Worth, Texas. I'm glad to be here today with my associate director, Dr. Maureen Spill, to provide a brief overview of the Evidence Center and then dig into today's topic, systematic reviews and related evidence synthesis methods. So thanks for joining us today. The Evidence Center was founded in 2022 as a scientific institute within the Texas Agricultural Experiment Station, otherwise known as AgriLife Research. The Evidence Center receives base funding from the state of Texas and conducts studies commissioned by local, state, federal, and international government bodies and other scientific and public health institutions. Now, the Evidence Center uses rigorous methods to synthesize existing evidence and answer scientific questions to inform decision makers of research, policies, programs, and regulations. We bring forward the state of the science with transparency on the relationships between agriculture, food systems, food and nutrition, and outcomes of human, environmental, and also economic health. The Evidence Center is here to help drive advancements in research methods and tools to bring speed and enhance the precision in synthesizing large bodies of evidence to bring transparency to the state of the science on a wide range of ag and food topics. Our mandate is to become a global leader and an independent, unbiased, and authoritative body for the review and synthesis of scientific evidence. Our mission is put into action following our 4 core principles: independence, rigor, transparency, and commitment. Here on this slide is a QR code that takes you to our webpage. On the Capabilities tab, you can review our wide range of capabilities. I'm going to introduce you to them at a really high level and then turn our attention to the area of focus for today, which is evidence synthesis methods. So what is evidence synthesis? It is bringing together existing scientific information in ways that can answer complex questions that no one study can answer, and in ways that describe the state of the science for a particular topic. It is a core function of the Evidence Center, and it includes the many different methodologies shown here and also encompasses a wide array of data analysis methods. However, the Evidence Center was also established to increase the precision and speed of current evidence synthesis methods, as well as develop new methods with a focus on those that allow us to consider the findings from different scientific disciplines and on the same or similar standards of quality. So to do this, logic models and frameworks allow the Evidence Center to create the scaffolding that is needed to evaluate multidisciplinary science in new and increasingly transparent ways. We convene and can facilitate expert panels and committees to provide balanced and transparent evaluation to inform decision makers, and we can synthesize not just the peer-reviewed literature, but also integrate other sources of food system and public health data to evaluate the potential impact of policies, programs, and regulations on their intended outcome. And I'm very excited about our scientific communications arm, the products we produce for our own work, and the services that we can provide for others. We drive to create exciting new ways to bring the science to life, to increase the rate of uptake and the rate of application of accurate scientific information. And last but not least, our mandate includes developing and facilitating scientific training to expand the use of rigorous evidence synthesis methods. We are established to develop training specifically to a policy or regulatory need or to the need of a research team, and to directly train those team members or new members as they join the research policy team. And today we're going to focus on our bread and butter, evidence synthesis. You have a treat to hear from Dr. Maureen Spill, our Associate Director. So with that, I'm going to turn it over to Maureen. Thank you.
Thank you, Kelly. All right. Evidence synthesis is a family of research approaches that systematically compile and draw meaning from bodies of scientific evidence. In this presentation, I'm going to cover different terms and types of evidence synthesis products, explain their purposes, and walk you through some real-world examples. There are a number of organizations that conduct evidence synthesis and have been leading method development. I'll be referring to several of those organizations throughout the presentation, and afterwards we'll have a handout available with some of these resources on our website. It turns out there are over 1,000 terms that have been used to describe evidence synthesis. This is actually the title of a recently published scoping review from Danielle Pollock and the Evidence Synthesis Taxonomy Initiative. The paper, published in BMJ, included this word cloud, illustrating all 1,000 terms and their relative frequencies. But to simplify, evidence synthesis can be organized into two broad categories. JBI, formerly the Joanna Briggs Institute, presents evidence synthesis as systematic reviews and scoping reviews. The Collaboration for Environmental Evidence uses the terms systematic reviews and systematic maps. Scoping reviews or systematic maps have also been referred to as big picture reviews. At the Evidence Center, we tend to use the term scoping review, so I'll stick with that for the rest of this presentation. In this webinar, I'll describe how evidence synthesis terms relate to these two categories. And since we don't have time for all 1,000 terms, we'll cover the more common ones. To start, I want to mention that there are common types of reviews that don't belong in the evidence synthesis family. A recent paper by Barbara Nussbauer-Straight, published in the Journal of Clinical Epidemiology, explains that literature reviews, sometimes called narrative reviews, typically rely on authors' subjective judgment to select and interpret studies. They lack formal, systematic methods that minimize bias, which is critical to evidence synthesis. Therefore, literature or narrative reviews should not be considered evidence synthesis. So getting back to the types of evidence synthesis with systematic reviews and scoping reviews, let's start by talking through some key features of each. First, it's important to establish that they have different aims. Systematic reviews have a focused aim to answer a specific question using all relevant evidence, which we call an evidence base. Systematic reviews have been used for decades in fields like education and medicine. They're often used to support evidence-based medicine and clinical guidelines with a focus on interventions. But systematic reviews are also used across many other disciplines to support decision-making in fields like social and environmental sciences. With that in mind, here are a couple examples of types of research questions that systematic reviews can address. Just keep in mind, this is not an exhaustive list. Systematic reviews may answer research questions about the effectiveness of an intervention or how one intervention compares to another. These are sometimes called effectiveness or comparative effectiveness reviews. But an intervention can also be a policy, an agricultural practice, or a change in the way a program, like a nutrition assistance program, is implemented. A systematic review may also answer research questions about the association between an exposure and an outcome. We use the term exposure when the independent variables are observed rather than assigned. For example, an environmental exposure could be observed levels of pollutants in the air, or a dietary exposure could be levels of a particular food consumed by a specific population. Systematic reviews may answer questions about the prevalence of a condition, called prevalence reviews, or the accuracy of a test or a measure, which are called diagnostic test accuracy reviews. Because they have a focused aim, systematic reviews are often used to inform decisions. On the other hand, the aim of scoping reviews is more exploratory, usually with the objective of describing an evidence base. They often are used to inform future research because they help identify key concepts, definitions, methods, and knowledge gaps. Given their different aims, systematic reviews and scoping reviews address different questions that result in different types of answers. Systematic reviews ask focused questions that result in focused answers. They provide information about the certainty or the level of confidence in an answer. For example, the systematic review question, what is the relationship between exposure to lead during pregnancy or lactation and concentrations in human milk, can be answered. Exposure to lead during pregnancy or lactation was correlated with concentrations in human milk with moderate certainty due to strong, consistent, and statistically significant correlations across studies, but high risk of bias. Scoping reviews ask exploratory questions that result in descriptive answers. For example, the scoping review I mentioned earlier about the terminology of evidence synthesis asked, what typologies, taxonomies, classification systems, or compendia have been proposed for categorizing evidence synthesis methods? And the descriptive answer found that there are more than 1,000 terms. They described the various facets used to distinguish them from one another and the fields of study they're used in, and so on. Now let's talk about the research process. Systematic review and scoping review processes start in similar ways. A systematic review begins by pre-specifying the methods that will be used. These are written out in a document called the protocol. In addition to defining the key components of the question, like the population, intervention or exposure, comparator, and outcome, the protocol specifies the search strategy that will be used to systematically search for all the relevant evidence. The eligibility criteria, which are a set of inclusion and exclusion criteria that get used during screening to systematically identify all the relevant evidence from the search results. Here, I'll point out that some of the terms that get used to describe evidence synthesis have to do with the types of studies that get included. You can include only primary studies with quantitative findings or only studies from qualitative research, called a qualitative evidence synthesis. You can include both quantitative and qualitative research, which is called a mixed methods review. You can have a systematic review of existing reviews. These are called overviews of reviews, or sometimes referred to as umbrella reviews. The protocol specifies what data gets extracted, a plan for critically appraising the evidence, which includes appraising the risk of bias of individual studies and the certainty of the body of evidence, and a synthesis plan. Similarly, scoping reviews begin with the development of a protocol that includes defining the question, like the population, concept, and context, the search strategy, eligibility criteria, extraction fields, and the plan for synthesizing, or more so organizing the evidence. Scoping reviews generally do not critically appraise the evidence, so those details aren't necessary in a scoping review protocol. Next, you register the protocol. Many systematic reviews get registered in Prospero, although there are other public registries such as Proceed, a registry for environmental science reviews. Scoping review protocols should be registered as well. Open Science Framework, or OSF, is a common place to register scoping review protocols. For both review types, once protocols are registered, the search can be conducted, then screening occurs to identify the included studies. This is where the methods begin to diverge due to their different aims. Systematic reviews typically involve detailed and sometimes extensive data extraction. Since a systematic review aims to answer a specific question about an intervention or exposure, it requires extraction of the relevant results, for example, effect sizes. Additionally, systematic reviews usually extract study characteristics such as data about the study design, participants, information about the intervention or exposure, and assessment methods, because these are all important in providing context for interpreting the results. The systematic review includes a risk of bias assessment. That helps reviewers understand the risk of inaccurate results in each included study due to systematic errors. A systematic review synthesis is aggregative. question gets answered by aggregating or pooling results from all the included studies. Synthesis can be qualitative or quantitative. Meta-analysis is a type of quantitative synthesis that pools the effect sizes across studies to get an overall effect estimate. Finally, a systematic review assesses the certainty of evidence or the confidence in the answer. This is important information for decision makers because it means a systematic review can not only answer the question, but it also describes the level of certainty around the answer. Since the aim of a scoping review is to describe the evidence base, data extraction tends to not be as extensive. Study results are generally not extracted. Instead, reviewers extract appropriate study characteristics to address the research question. Without results, there's no need for risk of bias assessments or certainty of evidence assessments. The synthesis in a scoping review is configurative, meaning it's more about the organization of the data. The scoping review question is answered by describing the evidence and including information such as the volume of evidence and evidence gaps. One type of scoping review output is an evidence or gap map. This is a graphical display of the evidence and may include data visualization in the form of a heat map or bubble map that show how many studies in the body of evidence have the characteristics described in the scoping review. Here's an example of a heat map that serves as an evidence and gap map. This one shows toxicant exposures down the side and outcomes across the top. For every outcome exposure pair, the color depicts the volume of research. White cells show gaps in evidence, and as the color gets darker, the volume of evidence increases. So the darker green cells have more research than the lighter green cells. This evidence map is static. However, there are interactive evidence maps. Here's an example, excuse me, this is an example of a dynamic interactive evidence map. Foods are organized in rows. Agricultural production processes are in the columns. The bubble colors indicate different heavy metals. The bubble size reflects the amount of evidence. When you hover over a cell, it'll show you the number of articles identified for that particular food process pair by heavy metal. You can expand the rows and columns to get more detail. In this screenshot, I've expanded the rows and columns for crops. Again, you can hover over a cell to see the number of articles. And if you click on a cell, you'll get a list of all the articles from that food process pair. This list is in alphabetical order, but you can sort the list by author or heavy metal. Systematic and scoping reviews may be conducted as new reviews, or they may be updates of existing reviews. When a review is started from scratch, it's called a de novo review. Systematic reviews can be updated. Once a review is complete, you can update the review by updating the search to identify new evidence and then follow the remaining steps of the process as outlined in the protocol, synthesizing the evidence from the initial review with the newly identified evidence. A more recent development is the living systematic review, where a review is initiated and completed, but then evidence is continuously monitored and incorporated into the review to allow for timely updates. This requires a detailed plan for the frequency and approach of integrating new evidence. It entails a long-term resource plan, both in terms of staffing and budget. But living reviews are particularly valuable for important questions with emerging research. You may have heard of rapid reviews. It's important to understand that these aren't just reviews that are done quickly by loading resources. They require less time because they compromise methodological quality. There's no one approach for rapid reviews. Methods are selected based on the specific requirements of each review. Rapid reviews do not provide the same rigor as systematic reviews, and therefore their use should be carefully considered as explained in Cochrane's Rapid Review Methods Guidance. Additionally, it's important to transparently report the methods used, highlighting the methods that were restricted and the bias that may have been introduced due to the use of those restricted methods. Okay, that was a lot of information. So now I'll take a couple minutes to provide some real world examples to help you understand the way these different types of evidence synthesis can be used to meet different needs. The first example is from a project we did on the role of seafood consumption in child growth and development, which was commissioned by the National Academies of Sciences, Engineering, and Medicine. We were commissioned to address two questions. The first was, what are the potential nutritional benefits associated with seafood consumption? For this question, the National Academies were concerned with seafood exposure during pregnancy, lactation, and/or childhood on nutrition outcomes in childhood. Our approach was to conduct 2 review updates using relevant reviews that were completed by the 2020 Dietary Guidelines Advisory Committee. The second question was very broad. It asked, what are the potential risks from seafood consumption? And the expert committee identified a long list of potential contaminants of interest, as well as a long list of health outcomes. So our approach began with a scoping review to inform our next evidence synthesis steps. Here's one of the evidence maps I showed earlier. All of the white cells are exposure outcome combinations without evidence, so they're not ready for systematic review yet. The cells in green do have evidence. Armed with that information, we were able to make informed decisions about our next steps. We completed 2 de novo systematic reviews, one on maternal-led exposure from seafood and child neurodevelopment, and a second systematic review with meta-analysis on maternal PCB exposure from seafood and child growth. Finally, given the amount of evidence on mercury and methylmercury, we conducted an overview of systematic reviews. Our overview included 29 existing systematic reviews on mercury exposure and child outcomes. The second example is a project related to assessing excess adiposity in children and adolescents. The World Health Organization is developing living guidelines for the integrated management of children and adolescents with obesity in primary healthcare settings. WHO undertook an enormous scoping effort with various stakeholders to identify questions of interest that were needed to inform the guideline development process. As you can see, one of the questions related to the diagnosis of obesity. The question basically asks, in children and adolescents, how accurate is BMI in diagnosing obesity? And is there a better method that can be used in primary healthcare settings? Our approach was to undertake a diagnostic test accuracy systematic review. I also want to mention that for this review, we gathered input from a large number of subject matter experts to help us develop the best protocol. In fact, we had input from 39 experts across 23 countries, and they helped us decide on the reference standards, index tests, thresholds for determining excess of adiposity, and the types of adiposity that should be considered in the review. I want to mention this because it's often a good practice to include expert consultation during protocol development, especially when the evidence will have a major impact, as it does here, in this case, clinical guidelines with worldwide reach. Next, we're going to pivot to 2 projects commissioned by the U.S. Food and Drug Administration on potential contaminants in foods. The first one focuses on contaminants in human milk and infant formula. We were given a list of questions from the FDA. We mapped those questions into this visual framework. Visual frameworks for families of related questions can help show the full scope of work and delineate parameters for each individual review question. We ended up conducting 6 systematic reviews that included examining the relationship between maternal exposure to contaminants and contaminant concentrations in human milk, contaminant concentrations in human milk and infant formula, There were also several questions on bioavailability, like bioavailability of the contaminants themselves, how contaminants affect bioavailability of nutrients in human milk and infant formula, and factors that influence this bioavailability. And finally, we also explored contaminant concentrations and biological samples from infants. My final example relates to contaminants and agricultural production of food from human consumption. This was also funded by FDA in collaboration with USDA. The research question was, what is the availability of peer-reviewed literature examining contamination and agricultural production of food for human consumption? As you can imagine, given the wording of this question, we undertook a scoping review. The scope was quite broad, even for a scoping review. FDA agreed to limit it to 4 heavy metal contaminants: arsenic, lead, cadmium, and mercury. But they didn't want to limit the foods included. So any food sold via interstate commerce was included. The production processes were limited to those prior to manufacturing. We had an amazing expert group that worked through a process flow for each food domain and delineated what process would be included as agricultural production processes, as opposed to processes that occurred later in the supply chain. We had almost 11,000 included articles. This is huge. So what do you do with that much data? We have nice tables showing the intersections of foods and processes and contaminants, but we wanted to go one step further to add value and make this data more useful for FDA, USDA, other food regulatory agencies and researchers and stakeholders generally. So we created an evidence map that I showed earlier. In closing, the Evidence Center was created to conduct high-quality evidence synthesis to inform decision makers of policies, programs, and regulations. And we want to be a resource to help you better understand evidence synthesis and to support you with your evidence synthesis needs. If you're with a government agency, you can find us on the GSA schedule. We hope you will connect with us. Our website will have a handout from today's webinar that you can download. Thank you.